All right, chapter 5, cellular metabolism. So cellular respiration. All right. This is actually one of my favorite things to teach. So hopefully I do a better job than usual. All right, so metabolism. So introduction to glycolysis and the lactic acid pathway. Okay. All the reactions in the body that require energy transfer, okay, can be divided into two things. So metabolism is the sum of all the chemical reactions that happen in the body. They can be broken down two big groups. Okay. Anabolism is the building up of stuff. Okay. Building up of stuff requires energy. Okay. That is counterbalanced by catabolism, the breaking down of stuff. Okay. Breaking down of stuff releases energy. Okay. Okay, so they drive each other, okay? It's not like you have one cell doing catabolism and one cell doing anabolism. It doesn't work that way. Like cells build and break down stuff all at the same time, all the time. Okay? Catabolism drives anabolism. So the catabolic reactions that break down glucose, fatty acids, amino acids, they build up ATP. The ATP is then used, right? They break down ATP to build up other stuff, okay? And it involves many oxidation reduction reactions, okay? The complete catabolism of glucose requires, in humans, it requires oxygen as a final electron acceptor, okay? It's called aerobic cellular respiration. I think you guys have gone through this in microbiology and stuff, right? So, okay. Breaking down of glucose requires many enzymatic catalyzed steps. The first which are anabolic. So glycolysis is anabolic, okay? Or sorry, not anabolic, anaerobic. So you don't need oxygen. So glycolysis, okay? There are some cells in the body that are just pure glycolytic. They don't require oxygen at all, okay? Okay, the complete breakdown of glucose requires 3 steps. Okay, glycolysis, which occurs in the cytoplasm, is anaerobic. It's by far the most ancient one of the 3. 2, the citric acid cycle, or the Krebs cycle. Okay, this occurs in the mitochondria as well as the electron transport. Okay. These 2 are aerobic, okay? They require oxygen to do. Like these 2 are the reasons why we have mitochondria in the first place, okay? So the complete breakdown of glucose, okay, is, so glucose, C6H12O6 plus, wow, there's a typo there. It should be 6. Oops. I also realized I didn't share my screen, so let me make sure I do that right now. Okay, sorry about that. I realized I didn't share my screen. Okay, so aerobic respiration of glucose, okay, occurs in 3 steps: glycolysis, okay, the citric acid cycle, and electron transport chain, okay. There's a typo right here. There should be a there should be a 6 right there. Okay, oops, 6 O2 goes to 6 CO2 plus 6 H2O plus ATP. Okay. Okay. Let me just go back and see the previous slides. You've probably just can follow this one. You should definitely know the definition between anabolism and catabolism. Okay. Okay. So here, okay, you have glucose coming in from the digestive tract or from the liver, and it goes into the blood plasma. We absorb it leaks out of the blood plasma into the extracellular fluid, where the extracellular fluid is then pumped. Glucose is purposely pumped into the cell cytoplasm, especially under the influence of insulin. Okay, and inside the cell, okay, in the cell cytoplasm, we go through glycolysis, okay, where it's converted to pyruvic acid, and then it's a major branch point, okay? If there is oxygen, it goes into mitochondria, undergoes the citric acid cycle and electron transport chain, and you spit out a bunch of energy. If you do not have oxygen, you go through anaerobic metabolism and you break it down to lactic acid. Okay? Okay, so the first step of glycolysis is, the first step of cellular respiration is glycolysis. Okay. Okay. So glucose is split into two pyruvic acid molecules. Okay. So a 6-carbon sugar is divided into two molecules of 3-carbon pyruvic acid, and you see the chemical equation here. Okay. Notice there's a loss of 4 hydrogen ions. Okay, so remember that means glucose is oxidized. Okay, so if something is oxidized, something has to be reduced. Okay, so these are used to reduce 2 molecules of NAD into NADH. Okay. Okay, so again, NADH, high energy molecule. We'll use that later. Okay. Okay. The net gain in glycolysis. So glycolysis is exergonic. So some of the energy is produced and used to make ATP. Okay. Okay. So there are 4 ATP generated, but there's a cost of 2 ATP. Okay. So 4 minus 2 is 2. So there's a net gain of 2 ATP. Okay. Okay. So now, okay, so here's how glucose is actually broken down. Okay, the first thing that happens to glucose, you add 2 ATP to it. You're priming the pump. Okay, the expectation is you prime the pump, you expect a bigger payoff at the end. Okay, and you do. So you spend 2, you get 4. That means you net gain 2. Okay. In addition to that 2 ATP you gain, you also gain 2 NADH, which is high energy molecule, and you'll see where the payoff is later. Okay. All right. And so you gain 4 and you gain these 2 NADH. All right. So the reactants and products. So the quiz is from the manufacturer and stuff, but usually this is what I focus on, the reactants and products. Okay. Okay. So here is an overview. So now this is where the split is, right? So I end up with 2 identical pyruvic acids. Okay. So when there is no oxygen to complete the breakdown of glucose, okay, we have NADH left. Okay. Now, because I have no oxygen, I have nothing for you to shove the NADH onto. Okay. So I take the NADH and basically I shove it back onto the pyruvic acid and I make lactic acid. Okay. This seems like an entirely wasteful thing to do, and it is. Okay. The reason is we usually have a different reason for NADH or different use for NADH. Okay, but we need NAD. NAD is not like in a large amount inside the cell. So we constantly have to, when we make NADH, the NADH has to go back to NAD very quickly so we can pick up more hydrogen from other places. It can't just sit there. Okay. All right. Okay, this is also called anaerobic metabolism or lactic acid fermentation. Okay, we already discussed this in microbiology, but yeast do a similar type of fermentation, except they break it down one little more step and they turn it into alcohol. Okay, you still have a net gain of 2 ATP. Okay, so you're still getting energy, and so muscle cells can survive for a while without oxygen by using lactic acid fermentation. In fact, in your body, and we'll probably go over this when we talk about muscles, you have some muscles, small muscles that are completely dependent on this pathway. Okay, they work faster. So glycolysis works faster. If you use glycolysis, you gain 5 times the speed. The payoff, however, is you get much less ATP. Okay, so things like eyelids, because the eyelid blinking doesn't require that much energy, but you need speed. You want to go through just the lactic acid pathway. Okay. Okay. Now, in most cases, okay, in most other cells, we have aerobic respiration. Okay, aerobic respiration begins with glycolysis, right? Now we can take the pyruvic acid and we can use it, we can break it down further. Okay, usually the lactic acid we make, we keep it as lactic acid for a while, and then once we're like have ample oxygen, we take it back to pyruvic acid, and then we finish breaking it down, and that is called paying the oxygen debt. Okay. 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 So the fate of the pyruvic acid. So the next step of the pyruvic acid is the pyruvic acid leaves the cytoplasm and enters the interior matrix of the mitochondria. Okay. There's one step called the intermediate step. Okay, where a carbon dioxide is removed from acetic acid. Okay, that carbon dioxide you breathe out. Okay, acetic acid is combined with an enzyme called coenzyme A to form acetyl-CoA. So remember, one molecule of glucose forms 2 pyruvates or pyruvic acids. Okay, so one molecule of glucose forms 2 molecules of acetyl-CoA plus 2 CO2. Okay, so there's a pyruvic acid, there's the coenzyme A, and now I'm attaching, okay, these 2 carbons to the S, okay, and I'm getting rid of this as CO2. Okay, this step I have again oxidized this side, I have reduced NADH or NAD into NADH. So again, I picked up another NAD. Okay, there's a bunch of names for the citric acid cycle. the citric acid cycle, the Krebs cycle, the tricarboxylic acid cycle. Okay, either way. Okay, the reason why it's called citric acid cycle because the first thing you make is citric acid. Okay. Okay, and the cycle because you literally go in a big circle. Okay. Okay. Um, okay. The important steps: you make one GTP, which is then converted to ATP. So you make one ATP per cycle, and remember you're doing this cycle twice because you have two acetyl-CoAs. So you pick up two additional ATPs per molecule of glucose. Okay, you pick up 3 NADs per cycle, which means you pick up 6 NADHs per glucose. You pick up 2 FADH2s per glucose. Okay, so there's the cycle. Do not memorize every enzyme and everything in this step because that is a huge waste of time. Okay, so for each glucose, this is what you get. Okay. Okay, just to help you a little bit. Okay. We also get from glycolysis 2 ATP, 2 NADH, okay, from transition step, we get 2 NADH. Total 2C2. Okay. So the total breakdown. Okay, so now let's look at carbon for a second. Okay, CO2, CO2. Sugar C6H12O6. All of it has been broken down. There is no more hydrogens. So it has been completely oxidized to 6 CO2. Okay, the 6 CO2 are what we breathe out. Okay. So total, we have 4 ATP, 2 plus 2 here, plus 2 there. We have two there, 6 there. Okay, so now we have 10 NADH. And we have 2 FADH2. Okay. All right. Oops. Sorry, I forgot to do the count for ATP. 2 here, 2 there, 4 ATP. So 4 ATP, 10 NADH, 2 FADH2s. Okay, so that's our count so far. All right. Oops. Sorry, I'm not having a good day today. Okay, so the electron transport chain. If you ever saw mitochondria, you'll see there's an outer membrane that looks kind of like kidney bean, and then there's an inner membrane that's all folded. Okay, that the reason why there are so many folds, and that's what makes mitochondria so successful, is they have an internal membrane that has a lot of folds. A lot of folds means there's a lot of surface area. A lot of surface area means I can shove a whole bunch of proteins onto that membrane and make it do stuff. Okay. Okay, what it probably does is something called the electron transport chain. Okay, the electron transport chain think of it as a molecular version of hot potatoes, okay? And of course, the potatoes are the electrons, okay? So I am taking the electrons and the hydrogens associated with the electrons, and I'm constantly passing them. Every time I pass electrons on, I reduce the next thing, and then it oxidizes by reducing the next thing. Okay? So that goes to that, goes to that, goes to that. Ultimately, where the electrons go is oxygen. Okay? The electrons get dropped onto oxygen to form water. Okay? Why that's important? This is your sole reason for breathing in oxygen. If you have no oxygen, right? If you have, oops, if you have no oxygen, then this doesn't work, this doesn't work, that doesn't work, that doesn't work. And eventually all you end up with is the glycolysis. Okay? So one more thing I want to show you. Okay, the NADH enters the cycle here. The FADH enters the cycle here. Okay, remember, so the FADH has less energy. There's two less electrons here. Okay. Okay. Uh, oops. Oh, there are all drawings out of there. Okay. Okay. The chemoosmotic theory. Okay. The electron transport fuels and proton pumps, which pump H plus from the mitochondrial matrix into the space between the outer and inner membranes. Okay, so NADH fuels 3 pumps, 1, 2, 3, and each pump brings a hydrogen into this intermembrane space. FADH2 enters at the second pump, okay? And therefore it only pumps 2 hydrogens in, okay? So you have 10 NADHs. Remember that chart? We have 10 NADHs, each pumps 3 hydrogens in. So for molecule of glucose, you're pumping in 30 H pluses. You have 2 FADH2s, each one pumps in 2, so you have 4 hydrogens in there, okay? So for a total of 34 hydrogens in that space, okay, per molecule of glucose. Okay, so I already explained the function of oxygen, okay? Okay, so there you go. Okay, so these hydrogens are pumped into space. They don't want to be there, okay? They want to go back to where they were, okay? First of all, there's way too many plus charges in there, and plus charges want to get out. The problem is they can't get out. Plus charges, charged molecules do not go well across that space, right? The phospholipid bilayer. The only way out is through this thing called ATP synthase. Okay, ATP synthase looks molecularly like a turnstile, and that's what it does. Okay, I know you guys have seen turnstiles for probably a year, but like the turnstiles at Dodger Stadium or Staples Center, you literally have to push that bar to get across. And that is what's happening here. The hydrogen, the only way they come back through is by pushing that bar on ATP synthase and getting through. Pushing that bar once generates one ATP. So you have 34 here, okay? So you're pushing 34 hydrogens back across, so you're generating 34 ATP, okay? Plus you have the 4 ATP from earlier, okay? So the theoretical ATP yield is 38, okay? Okay, the realistic yield is 30 to 32, okay? The energy is actually needed to move ATP out of the mitochondria in the cytoplasm. The other thing is that this isn't perfect. There are leaks in here and stuff like that. So, and there are enzyme costs. So roughly 30 to 32, okay? Okay, interconversion of glucose, lactic acid, and glycogen. Okay. So the cells can't store a lot of glucose. Glucose is not a good storage molecule. It is a very efficient, high energy, easy to move across molecule, but it's not an efficient storage of anything because water tends to, so if you look at molecular glucose, there's a whole bunch of OHs there, and all those OHs attract water around it. So like one molecule of glucose will have many molecules of water around it. So if you have many molecules of glucose, your cell would literally explode, okay? Okay, glucose is stored as a larger molecule called glycogen. Glycogen isn't nearly as attracted to water, okay? Okay. Glucose is stored as a larger molecule called glycogen in the liver and skeletal muscles, cardiac muscles. Okay, glycogen is formed via glucose with glucogenesis. Okay, glycogen. So the glucose is phosphorylated, isomerized, and then it forms basically a long chain. Yeah, I don't know where the chain is. There is one. Okay, the opposite of that is glycogenolysis. Okay, glycogen lysis, chopping up of glycogen. Okay, so whenever we need it, we break down glycogen in glucose again. Okay. Okay. So here you can see there's a pathway forward and back. Okay. Okay. Glycogen lysis in the liver. Okay. Glucose from glycogen is in the form of glucose 1-phosphate, so it cannot leave the muscle or heart cells. If your muscle is making glucose, then it's exercising and you don't want to lose that glucose. Okay. Liver has an enzyme called glucose 6-phosphatase that removes the phosphate so the glucose can re-enter the bloodstream and go to other cells. Okay. All right. The Cori cycle. Some lactic acid can be used in cellular respiration to produce carbon dioxide and water. Okay, again, we convert it back to pyruvate, and then we move it down the step again. Skeletal muscles make too much, so it's shipped to the liver. The liver converts it back to lactic acid, to pyruvic acid, NADH, etc. Okay. Okay. So the liver can convert the pyruvic acid back to glucose 6-phosphate. Okay. This can then be used to make glycogen or glucose. Okay. Pyruvic acid going to glucose is this thing called gluconeogenesis, glucose neogenesis formation of. Okay. 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 So here, okay, when we exercise, when we don't have enough oxygen, we make pyruvic acid, we make lactic acid goes to the blood, where it goes all the way back up, and it goes back to glucose, and it goes back into the blood, and back into the skeletal muscles, where it can be burned again, okay? Okay. We primarily burn sugar, but that doesn't mean we don't burn other stuff, okay? Lipids and proteins can be used for energy within the same pathways, okay? At some point, we run out of glycogen storage, okay? And when we run out of glycogen storage, we have to convert it to even more dense form of energy, and that is fat. Fat is even more reduced form of carbohydrate, so it's even higher energy per gram. Okay. Okay. So fat can be made of two components: the glycerol backbone and the fatty acids. And so this is where the glycerol comes from, and this is going to be where the glycerol is shoved into when you're burning fat, and the same for the fatty acids. The fatty acids always go to make acetyl-CoA, and then acetyl-CoA can be burned in the citric acid cycle, and eventually the transport chain. When we run a lot of citric acetyl-CoA, then we push it toward making fatty acids. Okay. So when we eat a lot of sugar, it goes this way and it makes fat. Okay. Okay. So ATP levels rise after energy-rich meal, and ATP actually inhibits glycolysis. Okay. So glucose doesn't complete glycolysis, and then acetyl-CoA is already formed together, and so you build lipids. Okay. Okay. So acetyl-CoA can go form cholesterol, which forms steroids and bile acids. They form fatty acids for triglycerides, okay, or phospholipids for cell membranes. Okay. Okay. So fat is stored as adipose tissue as triglycerides. Okay. That's a great way to store energy. 1 gram of fat is equal to 9 calories worth of energy. That is more than double the 4 that is in carbohydrates. Okay, and a non-obese 150-pound man, 80 to 85% of his stored energy is in fat. That's if you're obese, it's even greater than that by a lot. So fat, not only is it more dense because it's more reduced, it's also more dense because fat is repulsive to water. So all fat gathers together, and there's no water weight there. Okay. Okay. lipolysis, okay, is the breaking down of glycosides into fatty acids, okay, by using the enzyme lipase. Okay. Okay. The fatty acids can then be used and for energy as glycerol. Okay. Okay. So the beta oxidation of fat. So think of fat as a really long chain, and by really long chain, it's usually about 20 to 24 carbons long. Okay, so they're not showing all 24 carbons, but they are showing this. Okay, this is the end of it, and so this is the alpha. Okay, this is the alpha carbon, this is the beta carbon, this is the delta carbon. So alpha, beta. So beta oxidation, they cut the beta carbon, okay, and attach it to CoA for acetyl-CoA, and blah, blah, and then this is the same thing all the way through. Once you make acetyl-CoA, then you're going to take it all the way through these Krebs cycle and into the electron transport chain. Okay, so you cut 2 carbons at a time, 2 carbons, 2 carbons, 2 carbons, 2 carbons. Okay, and every time you cut 2 carbons, okay, you get something like 32 ATP. Okay, which is much higher. Okay. All right. Most of our fat is stored as white fat. Okay, but we have some brown fat. Okay, brown fat is primarily in newborns. Okay, newborns have this really small surface area to body ratio, or sorry, very large surface area to body ratio. So they can't maintain heat very well. Okay, so the brown adipose tissue, the brown fat, is used in thermogenesis. You're just constantly burning fat to generate heat, keeps them warm. Okay. Okay. One of the coolest ways that scientists are working on, they're trying to turn white fat into brown fat and go, okay, you're just burning more calories, because all brown fat does is burn fat constantly to generate heat. Okay. Okay. The sympathetic release of norepinephrine causes brown fat to form an uncoupling protein called uncoupling protein 1. Okay, so I kind of mentioned this earlier, but I didn't get too much into it, but here it is. Okay, uncoupling protein 1 sticks a hole in there. So instead of the hydrogen going through ATP synthase, they can run out of uncoupling protein 1. And since uncoupling protein 1 doesn't make ATP, you're just letting the hydrogen go for nothing. And that generates heat instead of ATP. Okay? It uncouples the electron transport chain from the ATP synthase. That's what uncoupling means. Okay? All right. Beta oxidation. So each acetyl-CoA can make that. Okay. So huge amount of ATP. Okay. Okay, ketone bodies. So when the rate of lipolysis exceeds the rate of fatty acid utilization, such as dieting, starvation, or diabetes, or the keto diet, the concentration of fatty acids in the blood increases. Okay, and that causes the liver cells to convert the fatty acids into acetyl-CoA and the ketone bodies. The ketone bodies and ultimately can go to different places, but the buildup in the blood can lead to ketosis. Okay. Proteins, are broken down into amino acids. Amino acids are usually needed from dietary proteins to replace the proteins the body uses up. We try not to actually use amino acids for energy. But desperate times, desperate measures. So excess amino acids can be used for energy or converted into carbohydrates or fat. Our bodies can make 12 out of 20 amino acids. Therefore, the 8 of them that we can't make are essential amino acids. We must eat them. That's what makes them essential. The others, we can make them from these 8 amino acids. We can use that to make the other ones. So here are the 8 essential amino acids. Pyruvic acid and several citric acid intermediates called keto acids can be converted into amino acids by adding the amino group. It's usually obtained from other amino acids. So you can make these guys from these guys, etc. I'm pretty sure you don't have to know exactly what enzyme does what. If there are more amino acids than needed, the amine group from glutamic acid can be stripped and excreted as urea. So if we remove the nitrogen, we need to get rid of it. We can't just have free nitrogen floating around because that ends up being ammonia. All right. Blah, blah, blah. So there you go. So we can transfer amino acids into this. So if we strip off the nitrogen, we add nitrogen to CO2, and we make urea, and then we pee out urea. So again, amino acids can go into the citric acid cycle, or the or here right above it into pyruvate, but we usually don't do that. The use of different energy sources. Glucose and ketone bodies come from the liver. Fatty acids come from fat, obviously. Lactic acid and amino acids come from muscle. So this is something I always wanted to explain. Different organs use different things. So brain, uses glucose primarily. When people ask me like, what do I do best? How do I do my best on a test? I'm like, eat a candy bar before the test. Your brain functions primarily on glucose. See how many pluses it has compared to other tissues? Your brain functions primarily on glucose. The only time it doesn't function on glucose is when you don't have any glucose, like if you're on a keto diet, and therefore it converts to feeding off of ketone bodies, and the gluconeogenesis your body generates to make a little bit of glucose from the brain. So your brain functions on glucose. Skeletal muscles when resting function on glucose, fatty acids, and ketone bodies. The liver can process a little bit of lactic acid. That's in the heart. but mostly, again, fatty acids. Okay? All right. I know that was kind of long, but I hope you guys understand it and enjoy it. All right. And this